Good morning, good afternoon, good evening, everyone. Thank you so much for joining us from various parts of the world that you're joining today's webinar. Before we begin, it's my pleasure to introduce to you my colleague, Dr. Katya Nikolov, as a speaker for today's webinar. Katya is an experienced physician scientist with over 10 years of academic and industrial experience in cancer biology and cell imaging. She has a background in solid tumor models, tumor microenvironment, and immunotherapy. In some of her previous roles, she led the development of 3D microtumor assay based on cancer biopsies to predict patients' responses to therapy and to improve outcomes. Here at Protein Fluidics, she's a senior application scientist. She's optimizing various automated 3D cell-based assays using the PUMA system, along with working closely with researchers and customers. Katya got her MD from the Russian National Research Medical University and did her postdoctoral research at Memorial Sloan Kettering Cancer Center before her various academic and industry roles.
Okay, well, thank you for the introduction, and thank you everyone for joining us at this webinar. Today, I will talk about automated organoid assays in Matrigel using the microfluidic-based PUMA system. But first, I'd like to acknowledge our collaborators. We work with Dr. Yoon-Young Choi's lab at Vanderbilt University, and today I will share some data that she kindly provided to us, and we'll show it to you today. We also work closely with Molecular Devices and Oksana Serenko and Matthew Hammer, who helps us a lot to acquire images that, again, I will share with you today and to do image analysis. So today I will do a brief overview of organoid models and different types of extracellular matrices. I will talk about PUMA system and explain how it works. I will share with you and walk you through the workflow that we have developed for Matrigel-based organoids to use in PUMA system. And I will show you a few applications in various organoids models that, again, we use with PUMA system. So when I was looking for the most, I would say, appropriate and the most accurate definition of organoids, I came across a very interesting review, and one of the authors was Mina Bissell. And they say that organoid definition lost the precision. And indeed, if you talk to different scientists, the way they define organoid would vary a lot depending on the subfield that they're coming from. But generally speaking, when we talk about organoids, we think about 3D self-organizing structure, which is generated from embryonic, adult, or pluripotent stem cell. Using particular supplements and culture medium and extracellular matrix, organoid cultures model specific organs containing multiple cell types, and they display physiological and cellular traits of the primary organ. And in recent years, we have seen really great advancement in establishing organoids, modeling various organs like intestine, gastric cancers, liver organoids, brain organoids, ovarian, and many others. So organoid culture typically requires growth factors and supportive matrices. The most used matrix for organoid culture is a basement membrane extract known as Matrigel. It is murine-derived extract containing collagen, laminin, and intactin. And although it is pretty widely used, I would say around the globe for the 3D culture, scientists do acknowledge its limitations, and efforts are being made to generate an alternative matrix that would be chemically well-defined, tunable, and reproducible. Now, there are different kinds of extracellular matrices that are being used in 3D research. For example, collagen is the most abundant fibrous protein in extracellular matrix. It provides support. It regulates cell adhesion, support hemataxis and migration. It can also be used at different concentrations to control for stiffness if it is required. Another option, for example, is to use decellularized ECM. They are based on removal of cells from tissue while leaving behind an extracellular matrix scaffold. Now, this type of ECM has really great potential because protein composition and ultrastructure of the native ECM is preserved. However, still work has to be done to adjust and improve biomechanical properties of such hydrogels, and as well as to improve their reliability and reproducibility. Now, there are other different kinds of native ECMs also used in research, for example, fibrinogen, alginate, gelatin, and of course, there's a number of synthetic materials that have been used to construct simple scaffolds for preclinical models. The advantage of such engineered scaffolds is that biomechanical properties of them can be tuned. They can be functionalized in many different ways. They are well-defined and more reproducible than natural ECMs. And these factors are particularly very important when you talk about getting reproducible and consistent results in preclinical models. Now, organoids are becoming one of the mainstream cell culture tools in many biomedical fields, and they're being used more and more in many different areas, including disease modeling, development, precision medicine, toxicology, drug discovery, and many others. And at Protein Fluidics, we have developed microfluidic-based Puma system that can help scientists in a variety of organoid-based applications. So why we have decided to develop such type of technology? These days, we witness tremendous shifts toward a more physiological 3D models in disease modeling and drug development. And typically, when we think about screenings and drug discovery, the first thing that pops into mind is high-throughput screenings. And usually, these screenings focus on large number of samples, but measuring only few parameters and produce low number of data points. Now, on the other side of the spectrum, we see there are many interesting, sophisticated, and interesting organ-on-a-chip devices. Typically, they would use 1 or 2 samples, but they allow you to produce really large number of data points. So essentially, what's happening here is that we move from very simple models to a much more complex model. And we think that the Puma system that we have developed fits really nicely into this middle spot where you would like to perform your assays on a medium scale, and yet it will allow you to collect substantial amount of data to do what we like to call multifunctional profiling. Really, our vision is to create open lab on a chip, which where you can use and maintain physiological conditions. It can use different types of organoids with or without matrix, depending on your assay. This organoid could range in size and be between 100 micromolar and 1 millimeter. And we automate all assay steps for you, which allows you to spend less time running complex assays, to use less reagents, minimize handling errors, and to get reproducible data with precious organoid samples. Our system is compatible with different kinds of organoids. It could be spheroids that are developed from established cell lines. It could be primary tumor-based tumoroids, and of course, different kinds of organoids with the matrix, and particular organoids cultured in matrix will be the focus of our discussion today. So here I show you a variety of examples of applications that we've been used with Puma system. We have demonstrated the utility of our technology for doing compound screenings and toxicity testing. We have done an interesting work with colleagues in diabetes field looking at the secretory profiles of pancreatic islets. We've done metabolomic studies. Recently, we presented another set of interesting data that we acquired together in collaboration with Tulane University in breast cancer patient-derived tumoroids. And here on the right side, I specifically highlight models that we've been using with Matrigel, and we run assays in Matrigel in our flow chip. So here is mapped out the workflow that we have developed for use with the Puma system. The first step typically would be a loading of the flow chip with organoids or spheroid, depending on the model that you use, and all the reagents necessary for the assay. Then the flow chip will be loaded into this device. The device itself is sitting inside the cell culture incubator. So essentially, you run all your assays using standard cell culture conditions in terms of oxygen and CO2 concentration and humidity. The next step would be to select the protocol. They are uploaded in the machine. And once the assay is done, your plate is ready for the analysis. And it can be used for fluorescence-based imaging. You can use different types of microscopes or high-content imagers. It can be used for luminescence readout using plate readers, or you can collect samples if necessary for the downstream analysis, for example, for metabolomics profiles. Now, the heart of the system is the flow chip, and here you can see the frame with 4 flow chips in it, and you can use between 1 and 4 flow chips, depends on the configuration of your assay. The dimensions on the frame and the flow chip resembles a standard 3T4 well plate, which makes it absolutely compatible with any type of instruments, like different kinds of standard plate readers, microscopes, or again, high content imagers. Each flow consists of 8 test lines, and each test line is designated to 1 sample. You can see here that in the middle of each test line, we have a well where we call it sample well, where your organoid is positioned. And then this sample well is connected to a number of reservoir wells through the microfluidic channels. These reservoir wells can be filled up with compounds, with wash buffers or lysis buffers, or different types of medium, conditional medium, for example. It's really highly customizable depending on the configuration of your assay. Now, the well itself is pretty small. The volume of it is 20 microliters, and you can see here that the organoid is located at the very bottom of the well in the protected sample chamber, which is our proprietary technology. The dimensions of the protected sample chamber at the bottom diameter is 1.2 millimeters. This sample is very well protected, and there is no real risk to lose it or break it during your assay. The optical bottom of the flow chip is suitable for high resolution imaging. And these channels on this side allow for fluid exchange. We can do up to 95% fluid exchange in the well. However, if your assay requires to completely drain the well, you can do so as well using a special channel at the bottom of the flow chip. Now, because, like I said, organoids typically require matrix, we have developed a protocol to manipulate and perform assays in Matrigel-Grown organoids. And again, it consists of the steps where we first retrieve organoids from the Matrigel and transfer organoid to the flow chip and then run our assay. And I will talk about more specific details in the next slide. What I would like to point out here is that although this protocol is developed for Matrigel, our system can be used with different kinds of hydrogels. And we've been working also on optimizing protocols for other hydrogels as well to be able to use them in our system. So here, I am showing you in details the workflow that we have developed for Matrigel-Grown organoids. Our flow chip can be used with Matrigel at concentration between 20 to 30% without any impact on fluid transfer and performance of the flow chip. Now, typically, scientists would culture organoids in 90 to 100% Matrigel dome. Therefore, we have developed a procedure to retrieve these organoids using cell recovery solution from the dome. And then we need to transfer them to 25 to 30% Matrigel. You can use either Eppendorf vial for that or different kinds of plate, again, depending on the setup of your lab. And then what we need to do is aspirate this pick up these organoids in really small volumes. So in our lab, we use a volume between 3 to 4 microliters, and it works very nicely. And the reason for that is to have your organoids positioned at the very end of the tip. And that makes it really easy to dispense them into the flow chip. Now, here on the picture, you can see that during the dispense of the organoid in the flow chip, Matrigel forms sort of a mini dome inside the protected chamber, which is very convenient, and your organoids are suspended inside this dome. And then we'll let Matrigel solidify and put 20 microliters of culture media on top of it. So now when your sample in Matrigel and organoids are loaded and all the reagents are in the flow chip, you're ready to start running your assay. The interface of the system is very simple and easy to use. All you need to do is just to select the protocol from the list that is already preloaded in the machine. All you do is just click this run button and can walk away, do any other things, because all the automatic steps of the assay will be happening inside the machine for you. Now, let's take a closer look what's happening inside the flow chip during run of the assay. So here you can see schematically how the flow chip looks like with this sample inside it. What we can do, for example, is to first drain the old medium to waste and then transfer fresh medium, incubate your organoid for the desired amount of time, drain the sample, and change it for compounds. Incubate organoid for, again, appropriate amount of time. We can collect supernatant, for example, for analysis of secretion factors, transfer staining solution. Once staining is done, we remove it, and you can wash your sample if needed. Now, at this step, your protocol is completed, and you can proceed for the downstream analysis. Now, in the next part of the talk, I would like to talk about few specific applications using various organoid models that we've been using with a Puma system. And we will start with lung organoids. For lung organoids, we use exactly the same workflow that I just described in detail, so I will not spend too much time on it. And here I would like you to show an example of how organoids look like inside the flow chip in Matrigel. You can either use a single organoid or multiple of them. They are kind of suspended inside the Matrigel and positioned in different level. And here's an example how with our Z-Stack, we were able to capture 4 of them. You can see that they very well maintain their shape. They are very well protected in the chamber. They pretty well stained. And I'd like to point out how nice these images look like and how great the quality is, so that you can really identify every single cell as a separate object. And that is very important for the downstream image analysis and to get high quality data. Now, here's another example of lung organoids. They were stained with Hoechst calcineum and mitochondrial stain. And you will see here 2 organoids, again, located at different planes. Again, they're very nicely and round shaped. You can see that they're hollow inside. And yeah, they look very beautiful. Here, I would like to show you an example of drug treatment response assay that we've done in lung organoids. These organoids were treated with the drug ibrutinib for 48 hours, and then they were stained with a tedium homodimer to detect dead cells, and with Hoechst to then calculate the amount of viable cells. And again, you can see here that the quality of image that we were able to acquire, even using Matrigel, is pretty high. It allows to identify viable and dead cells very nicely, and it's a very clean image. The analysis was done here using MetaExpress software, And then this data is translated into this plot where you can see that clearly upon exposure to the treatment, we can see substantial cell death and substantial cell killing compared to control, untreated control. Now, I'd like to share with you another example. Here we'll talk about intestinal organoids. Again, we use similar workflow that I described today. These organoids were formed from isolated intestinal crypts. They were also cultured in 100% Matrigel dome. Then they were transferred to 25% Matrigel, transferred to the flow chip, and we've done staining for the viability and imaged using confocal system. And here I'm really, I would like to share with you these beautiful images. Let me, something, okay, working. So you can see how beautiful it is, and you can really see clearly all these structures of this organoid. It's stained very nicely. So again, I cannot stress enough how important to get high quality data, high quality images for the downstream image analysis, so that you can get high quality data, quantifiable data out of it. And you can see that with Puma system, even with Matrigel, we have the capability in automatic way to get such kind of images and data. And now I'd like to share with you a set of data with gastric organoids that was kindly provided to us by Dr. Choi's lab at Vanderbilt University. The focus of Dr. Choi's group is to understand the oncogenic roles of KRAS activation in gastric carcinogenesis. Her group recently established novel mouse metaplastic and dysplastic organoid lines, which really nicely recapitulate the phenotype and characteristics observed in the gastric carcinogenesis cascade, similar as they are seen in mouse model and in human gastric cancer. So in their research, they use mouse model with active KRAS expression in the stomach cells, and they create organoids by isolating glands in gastric tissues, and then they transfer them to the Matrigel and for the formation of 3D structures. And here you can see this image showing actually the formation of around 3D structures over time in Matrigel. Here's outlined the workflow that they have been used in their lab. Again, they start their culture with 100% Matrigel, then they transfer their organoids to 25% Matrigel. They keep them for about 3 days before they start running assays. And then they, using inverted microscope, they have established technique to pick up these organoids and transfer them to the flow chip. Here you can see that sitting in the flow chip, organoid maintain their integrity. It has very nice round shapes and maintain morphological features. And then these organoids are used for different types of analysis, for example, staining with compounds, and then staining for viability markers or specific markers depending on the goal of your assay, as well as samples can be collected for example, analysis of secreted markers in these organoids. Now, using this established gastric organoid model, they examined how cellular behavior changes by inhibition KRAS signaling. And in this study, organoids were treated with the MEK inhibitor, which is a downstream mediator of KRAS signaling. And here, first, I show you these beautiful images. This is an example of organoid which was stained with calcium to detect viable cells and with ethidium homodimer to detect dead cells. You can see here that a treatment with MEK inhibitor results in substantial cell death in these organoids. And again, what I would like to highlight here is how beautiful these images, how crisp and high resolution these images are, which really provides you a lot of information and allows you to quantify them to get high quality data for the analysis. what they also done, they stained organoids with antibodies against intestinal enterocyte apical membrane markers. And here I would like to show you first staining that was done using standard paraffin sections, and to compare it with the staining that was obtained using automatic protocol in the flow chip and then imaging with microscopy. First of all, you can see obvious changes, morphological changes, and changes in the markers expression upon inhibition with MEK inhibitor. And these phenotypic changes and markers indicate regression of these plastic structures and behaviors. You can see that the shape of the organoid upon upon exposure to MEK inhibitor has very nice and round shape, and with the thin epithelial layer. But interestingly, what I also would like to point out here is how concordant these images are between flow chip and paraffin sections. Now, those who ever done embedding and staining of organoids or any 3D structures know very well how cumbersome and difficult this process is. It requires a lot of different kinds of reagents, different instruments, whereas when you use Puma system, there's really no need to do such a heavy processing of your sample with the risk to break it or to lose it. You really keep your sample in a very safe and physiological environment, and it allows you to stream and automate all the steps of these complex protocols, again, use less reagents, and at the end to get these nice and beautiful high resolution images for the further downstream data analysis. And to wrap up, I would like to show you in general how we approach multifunctional profiling. This particular study was done using breast cancer patient-derived tumoroids. We treated them with different compounds. And what we've done, first we stained them with viability stains to assess viability profile of these organoids using confocal imaging. Then on exactly the same sample, we collected sample supernatants to process them for the downstream metabolite analysis, as well as we've done immunofluorescence staining to detect specific markers and again do image quantification and analysis. So you can really see that in a very simple way and in very protected way, you can use your organoids to generate really large amount of data and on the same sample. So very convenient and easy to use. And so to summarize what I've talked today, I hope I convinced you that Puma system is a really unique technology, open lab on a chip system that allows you to do different types of assay and create different types of workflows for your precious samples. It allows to do multifunctional profiling and really collect large number of data for your samples. And what I've showed you today, our system is compatible with different types of extracellular matrices. Specifically, we talked about compatibility with 20 to 30% Matrigel, and I showed you a pretty robust workflow that we've created for Matrigel-Grown organoids from automated assays that you can further use in your work. And so I would like to mention here that you can go to our website and download this technical note, which has all the details you need talking about how to use Matrigel and how to run assays with Matrigel-based organoids. Please reach out to us if you have any questions about the technology itself, or if you'd like to talk about specific assays that you do in your research and that if you would like to know how to use them with a Puma system. And with that, I would be happy to take your questions.
Thank you, Katya. There are a bunch of questions here, and one comment I would like to quickly make. Michelle, you asked if the webinar is being recorded and will be available for viewing afterwards. Yes, we will make it available by the end of the week. It is being recorded. So some questions for you, Katya, are: Let me start with the first one. This question is from Terry. Does the 25% Matrigel solution form hydrogel, or does it remain as a liquid?
So it does form hydrogel, but of course it's not as stiff, I would say, as 100% Matrigel. You still can pipette it. It's not as easy, I would say, as pipette liquid, but it's pretty, like, we have really robust protocol developed for that. It does form sort of hydrogel.
Okay. So one of the questions was also, does the Puma software apply the same protocol to four flow chips when put together in the machine, or is it possible to run 4 independent protocols at the same time?
No, we cannot run 4 independent protocols. Your flow chip, depending, again, you can use between 1 and 4 flow chips, but only one protocol can be run at the same time for the plate.
Okay. Another question was, have you tried using brain organoids?
I personally haven't tried that yet, but maybe Dr. Cromwell can comment more on brain organoids if they've done in the past. I know we've done some work on that.
Yeah, we've done some work with stematics microbrains, done some functional assays, and actually some of the data was published in our recent SLAS technology article. And then we also have other users working with brain organoids doing immunofluorescence staining of fixed organoids. The immunofluorescence staining protocols are particularly useful because of all the fluid exchanges you have to do with, blocking primary antibody, washing secondary antibody, et cetera. So, using the protected chamber to facilitate those transfers it gives you a lot of advantages.
So one question from Bernd is that, will the 25% Matrigel conditions affect the quality of the ECM and or perhaps affect the way the cells interact with the ECM Matrigel?
So I would answer this way. There may be some interferences and there may be some effect, but the way we see it is that you would use your standard culture conditions for creating and culturing organoids, generally speaking. And the assays that we run are not so long, so that's why it's provide all the necessary support and binding. It's still there, but it's not really, for that long that it would really, you start seeing, any differences or changes.
What other kind of readouts can you get from the organoids in the Matrigel?
We can use different kinds of readouts. Like I said, we can do stainings. It could be staining for viability. It could be staining for very specific markers. We can do collection of the samples and do secretion analysis. And right now, there's some ongoing work looking at metabolite secretion. So as soon as we have some interesting data, of course, we'll share it with you. So yeah, really different kinds of assays.
So I'm just looking to see if there are any further questions. And one more question that I want to ask you, Katya, is it possible to perform, say, like with spheroids, where you can do, cell titer glow assays, perhaps, and look at readouts, the luminescence readout in a plate reader? Can you do some similar kind of assays with organoids in Matrigel?
Technically, it is possible. We need to perhaps run some optimization, to make sure that lysis is complete in terms of this concentration of Matrigel. But yeah, 3D cell titer glow should be possible to do and to achieve full lysis of the structure.
Fine. So one question from Ori is, how does the system enable temperature exchange with an incubator?
Okay, I will start answering this question and then I'll pass it to Evan. So generally speaking, system is sitting inside the incubator and it utilizes the air that is the same, let's say, environment and the same air that is inside the incubator. But Evan can go in more technical details on that.
So the system just will equilibrate with the incubator temperature. It takes about 30 minutes for it to equilibrate once you put it in from room temperature. And then the gas exchange is also because of the pneumatic action we use to move fluids around, we're constantly exchanging gas from the outside, the incubator environment, to the top of the media. So you get natural gas exchange occurring to keep your media at 5% CO2, et cetera.
So if there are no more further questions, I would like to end the session, but we will stay online and take your questions by chat. But you could also reach out to us at our website, and feel free to set up an appointment with us, and we'll be happy to go over, the protocols again with you and discuss what are the best options for your team. All right. Thank you so much. Thank you, Katya. Thank you, Evan. And thank you all for attending our session today. And hope we will see you, and we hope to hear from you. Thank you.
Thank you.
Thank you, Ross.
Bye.
Thank you, Katya.